everyone. I'm going to talk about the throat culture lab. So in this lab, you were supposed to take a sample from your throat. So it was a cool lab, and you were going to swab it on a blood agar plate. So these red plates are blood agar. You are ultimately supposed to see what bacteria grow from your throat. I'm going to start off with a little bit of background before I talk about the lab. So in anatomy, the throat is called the pharynx, and your throat or your pharynx is a warm, moist environment, and there's a lot of bacteria there. So we say there's a mixed normal flora. So in someone's throat, as you can imagine, you see bacteria from their upper respiratory tract and their digestive tract. There's just so much bacteria there. The most common throat bacteria or pharynx bacteria that we see are those of the Streptococci species, but there's a lot of other bacteria that are present in a throat culture sample. Besides normal flora, the pharynx can also, your throat can harbor potentially pathogenic bacteria, and these pathogenic bacteria are usually kept in check by your normal flora or the good bacteria that's there. The pathogenic bacteria are usually the ones that cause things like sore throat during a cold. So in this lab, you are going to obtain throat swabs, so these things right here, just like a Q-tip, and you are going to get a throat swab from your throat, and you are going to streak the sample on sheep's blood agar. You are going to do a streak plate technique. So remember, that's when you're trying to isolate colonies with sheep streak technique, and we're streaking our throat sample on sheep's blood agar because it contains blood which is similar to our human body. So sheep's blood agar mimics kind of what you would see in the human body, and we're trying to grow organisms that we see in the pharynx. So this is a differential medium agar. It's a differential medium agar because it helps differentiate different types of hemolysis patterns, which we're going to talk about. And the bacteria that are in your throat, we can categorize them based on the hemolysis patterns. Hemolysis, lysis is to break, remember, and hemo is blood. So we're seeing how they affect the blood in the sheep's blood agar, so how they destroy the red blood cells. So you're going to take a throat sample, do a streak plate technique to isolate colonies. You incubate the plate, so you put them in the incubator for 24 hours, and then you come and see the results of what could potentially be in your throat culture or your pharynx culture. And remember, I said the most common bacteria that we should see is Streptococcus. So hemolysions are toxins produced by bacterial colonies that either partially or destroy surrounding red blood cells. And so there are 3 hemolysis patterns that we can see on your blood agar plate. You can see alpha hemolysis, beta hemolysis, and gamma hemolysis. Alpha hemolysis patterns are when you see partial clearing around where the bacteria grew, and you see some greenish pigment. So if you just focus here, if you come back and you look at your bacterial agar plate, you might see some alpha hemolysis. The way we know that it's alpha hemolysis is you see the bacteria grew, and around it you see a green pigment and some clearing areas. And alpha hemolytic patterns on blood agar indicate normal flora, which we all have. So all of our plates should have alpha hemolysis or alpha hemolytic bacteria, and the most common bacteria in the throat, which is Streptococcus. Some people may additionally see beta hemolysis. Beta hemolysis is when you do not see any more red blood cells. They're completely lysed. So you just see clearings everywhere, as if the bacteria just broke all the red blood cells. Beta hemolytic bacteria indicate usually pathogens. So if someone has strep throat, a lot of times when you take their throat culture and you swab it on the plate, you will see beta hemolysis patterns. And again, it's complete clearings around where the bacteria grew. You do not see red blood cells at all. So those bacteria are causing, imagine your cells here, to die off. And then gamma hemolysis, you see no clearing, you just see where the bacteria grew, you see dark colonies, dark bacterial colonies, and that's basically it. So with alpha hemolysis, you see a green sheen. This is the most common. Everyone's plate will have alpha hemolysis on it, and you see some clearing, and it indicates the normal flora. Beta hemolysis indicates pathogenic bacteria such as Streptococcus pyogenes, which causes strep throat. Remember, there's so many Streptococcus organisms. One of them is Streptococcus pyogenes, Staphylococcus aureus, and then gamma hemolysis, we just see the bacterial colonies group. So I know that this is alpha hemolysis here because I see partial clearing and a greenish pigment. I know this is beta hemolysis because there's clearing of all the red blood cells. And then if you look at this blood agar plate, here there's a complete clearing around this bacterial colony. So I know that this is beta hemolysis, which indicates usually pathogenic bacteria. And then these are all alpha hemolysis. So the objectives of this lab is I want everyone to know the most common throat bacteria, which is Streptococcus. All of us have Streptococcus in our throat. Know the name of the medium you used. You used sheep's blood agar, and you're testing to see different hemolysis patterns. Given plates, I want everyone to know what alpha hemolysis looks like, beta hemolysis looks like, and gamma hemolysis looks like. Alpha hemolysis, we see some clearing and a greenish sheen. So if you see colonies, it's alpha hemolysis. If you see, colonies and some clearing. Beta hemolysis, which indicates usually pathogenic bacteria, we see complete clearing of red blood cells. Gamma hemolysis, we just see the bacterial colonies. Both alpha and gamma hemolysis indicate normal flora in the pharynx or throat, and then the beta hemolysis indicates that someone could potentially have pathogens. So which one is pathogenic? Beta hemolysis. And possible pathogens are Streptococcus pyogenes, Staphylococcus aureus if it gets in your throat where it shouldn't be. And another objective is understand the concept of a healthy carrier and how this might impact healthcare workers. A healthy carrier is someone who carries the pathogen, but they're not sick. So maybe I don't have a sore throat and I do this experiment on myself. So I take a pharynx sample and I do streak plate technique and I come back the next day and I see complete clearing on my blood agar plate. So I see beta hemolysis, which is complete clearing, but I don't have a sore throat. This indicates that I might possibly be a carrier of a pathogen and not know it. I myself am not sick, but this could impact everyone around me. So this is why healthcare workers need to be careful because they work in healthcare and they're around immunocompromised patients. They could be carrying a lot of things that might not be harming them, but could be harming other people. So just keep these things in mind, and I hope that you guys kind of get the picture of what you would have done in this experiment if you guys had been doing it.